So my name is Wilfred Vanderdonk, and our lab at Illinois studies natural product biosynthesis, in part in the hopes of discovering novel enzymatic reactions. And our speaker today, Paige Daniel, has exactly done that. So Paige is a biochemistry graduate student. She joined my lab in the fall of 2017. And in the short 3 years or so that she has been in the lab, she's done a really phenomenal job to unravel a completely unanticipated biosynthetic pathway in which heterocyclic small molecules are made by post-translational modifications. And that's what she will be telling you about. And so, Paige, please take it away.
All right, can you guys all see my screen okay?
Yes.
A pointer going. All right, so thank you, Wilfred, for that kind introduction. And it's my pleasure today to talk to you about my project, which involves Pyrroloiminoquinone biosynthesis and how that relates to small molecule biosynthesis by post-translational modification. So like Wilfred said, our lab really focuses on novel biochemistry, and we really focused on novel biochemistry in the context of natural product biosyntheses. One of the large facets of research that our group covers are RIPs, which are ribosomally synthesized and post-translationally modified natural products. Some examples of these natural products include the Lanthipeptides, the Streptomyces, and the lasopeptides. And what makes these systems unique is the biosynthetic gene clusters that encode a precursor peptide that is modified by the biosynthetic machinery that is encoded within that gene cluster. The precursor peptide generally comprises an N-terminal leader and a C-terminal core motif. The N-terminal leader acts as a recognition motif for the biosynthetic machinery to grasp onto and modify the C-terminal core. After those post-translational modifications have occurred, then a proteolytic cleavage event generally occurs to release the mature RIP from the leader peptide to afford your natural product. The chemistry that I'll be talking about today was derived from our study of the class 1 lanthipeptides, which are a subclass of the RIP natural products. And so in the class 1 lanthipeptide systems, these systems have a LanB enzyme, and LanB enzymes have an N-terminal glutamylation domain and a C-terminal elimination domain. And what these domains are responsible for are glutamylation of serine and threonine residues that are encoded within the precursor peptide in these systems. So the LanB enzyme will glutamylate the serine and threonine residues and then also perform the subsequent elimination of glutamate to afford dehydroalanine and dehydrobutyrene residues. These modified residues can then undergo intramolecular Michael-type additions with thiols that are also encoded within these precursor peptides to generate the macrocycles, the methylanthionine and lanthionine macrocycles that denote this class of natural products. One such natural product is nisin, and you can see that in this structure that I've shown, there are 5 macrocycles that were installed on this natural product. In our studies of the type 1 lanthipeptides, we performed large bioinformatic analyses to study these enzymes, and this is how we ended up finding the enzymes that I'll be talking about today. So when we do a simple blast search of NISB, which is the enzyme responsible for nisin biosynthesis, you're going to pull out several sequences for which the glutamylation and elimination domain are encoded as one bifunctional protein. But we also pull out sequences for which the glutamylation domain and the elimination domain are encoded as separate proteins. What we additionally pull out are the sequences for which the elimination domain is not encoded within the biosynthetic gene cluster, the operon, or the organism altogether. So we decided to investigate these sequences to see if they may have some alternative function than their split and full-length counterparts. When we do a sequence similarity network analysis, which compares the similarity between the sequences, we find that the small LanBs actually separate into a clade separate from that of the full-length LanB counterparts. This indicates to us that these might have some alternative function. So we decided to probe where these sequences were in the organisms and look at the biosynthetic gene clusters that contain these sequences. And what we find is that up to 7 of these sequences can be found in one biosynthetic gene cluster. And this is atypical, as LanBs are generally found 1 per biosynthetic gene cluster or operon. We decided to look at this model cluster in Pseudomonas syringae to find out what these enzymes were doing and if indeed they did have some alternative function. What we found was that when we co-incubate TGL-A, which is the precursor peptide associated with that cluster, with TGL-B, the small LanB, we find that it does indeed modify the peptide. But this mass shift is not indicative of glutamylation, but instead it is indicative of cysteine appendage. And when we investigated the natural product that was produced by this transformation, we found that it's actually appending cysteine to the C terminus of the ribosome in an ATP and tRNA dependent fashion. So in these systems, the C terminus of the precursor peptide gets phosphorylated with ATP. We can then have tRNA come in to attack the carbonyl and eliminate the phosphate group. You can then hydrolyze the tRNA, and now you have cysteine appended at the C terminus. At this point, you might be wondering, why would a system make a protein to add an amino acid that could otherwise be genetically encoded? In our further studies of the system, we found that the tailoring enzymes within that cluster generate a series of phenomenal reactions, including beta carbon excision of the cysteine molecule and carboxymethylation, and then a proteolytic cleavage event to free the modified amino acid from its precursor peptide substrate. And we generated this 3-thioglutamate molecule. Then the precursor peptide can then be recycled for the next catalytic turnover. So in these systems, you recycle the precursor peptide and use it as a catalytic scaffold to generate large quantities of a small molecule derived from an amino acid. Because of the unique chemistry that these enzymes perform, we've decided to coin them peptide aminoacyl tRNA ligases. After our investigation of this model system in Pseudomonas syringae, we decided to pursue other clusters that contain these peptide aminoacyl tRNA ligases. So this is where the focus of my talk today will be on in this Ammosamide biosynthetic gene cluster from Streptomyces species CNR 698. So this biosynthetic gene cluster was reported in 2016 by Moore and colleagues. And what they reported was that this biosynthetic gene cluster is responsible for ammosamide biosynthesis, but they were unable to determine where the tryptophan that this molecule is derived from came from. They anticipated that this tryptophan here at the C terminus would be the natural product source of tryptophan. But when they mutate this tryptophan to any other residue or delete the tryptophan altogether, the production of the ammosamides is not abrogated. They had annotated these proteins as lanthipeptide dehydratase-like enzymes. So indeed, they look like they could be doing some function similar to lanthipeptides, but unbeknownst to them, they were actually indeed pearls. So when we look at this sequence now, we see that these are pearls, and we anticipate that one of these pearl enzymes is actually going to be the source of the tryptophan. And we conducted a series of experiments truncating the peptide to see if one of the pearls would add the tryptophan here, but nothing seemed to work. And so we decided to compare the sequence of the precursor peptide from the ammosamides to other precursor peptide sequences that share high sequence similarity. And what we find is that some of these precursor peptides have an extension sequence at the C terminus, and other sequences terminate in this C-terminal alanine. We hypothesized that potentially one of the 2 types of proteases encoded within the biosynthetic gene cluster might be responsible for the cleavage here, and that indeed the tryptophan would be added to the C-terminal alanine. So we truncated the peptide, removing these amino acids at the C terminus, and generated a new peptide, AMMA star. And indeed, when we co-express AMMA star with each of the pearls, we find that one of the pearls, AMMB2, will now append tryptophan to this modified peptide, AMMA star. So now we know where the tryptophan is derived. But there are still 3 pearls left in this system. And that's a bit puzzling because, as you can see, 1 tryptophan seems sufficient for the production of the ammosamides. We also don't know where these amino groups come from, but it is my pleasure to tell you today that we do know a little bit more about what the pearls are doing and where those amino groups are coming from. After we identified the product of AMMB2, the tryptophan appended at the C terminus, we decided to investigate the other sequences within the biosynthetic gene cluster. And what we found was that AMMC will modify AMMA star trip, that modified peptide with the C terminal tryptophan. And in Brad Moore's studies, he was able to identify that AMMC was an essential protein, but he couldn't figure out what that modification was or what the product would be. And so we did this analysis and found that AMMA star trip is modified by AMMC and is trihydroxylated by this enzyme in an FMN-dependent manner. We corroborated this finding with high-res mass spec to confirm that the modification was to at least to the C-terminal fragment. And we predict that this is the structure that we now observe, this triphenolic species. And we predict that this is a structure based on downstream NMR of a later intermediate. Another observation that becomes important is that over time, this plus 48 Dalton product degrades into a plus 46 Dalton product. And we also observe a color change. So this is indicative to us of some oxidation to potentially a quinone. And the peak broadening that we observe here is generally indicative of quinone formation. And this particular structure becomes important for the next catalytic transformation. So now that we have our product, the triphenolic product and the quinone product, we went back to see if any of the pearls would now modify this substrate. And we found that AMMB3 does indeed modify the peptide. But what we observe is a plus 11 Dalton shift. And there is no amino acid that you can add that would give an amino or a mass shift of plus 11 Daltons. So this was a bit puzzling to us. And we were able to corroborate that it was indeed a plus 11 Dalton shift to the C-terminal fragment. But we needed to figure out what this 11 Dalton shift was. Thankfully, I had been working on an orthologous system, and this ortholog actually shed light onto what the AMMB3 product was. So in this orthologous system, we have a system in which we have a C-terminal alanine and tryptophan, just like in the ammosamide system. And we also have a trihydroxylating enzyme, and we also observe the same color change and quinone formation here. And then we also have the AMMB3 ortholog, which modifies the BHAC1 product. And we observe now a small blip at plus 57 Daltons, which corresponds to the appendage of glycine. Surprisingly, when we conducted NMR on this product, we found that the glycine was actually appended to the R group of the modified tryptophan rather than to the C terminus of the peptide. And this was exciting because now we can rationalize why we have this quinone. This vinylogous acid here is primed for the same reactivity as the C-terminal carboxylate. So we can add on glycine now at the R group. And now we can make sense of the other masses that we observe. So when you have your quinone product that gets modified by AMMB3, you would phosphorylate the vinylogous acid, and then you would eliminate the phosphate group and hydrolyze your tRNA to afford this glycine conjugate. And this is what we observe via NMR. But this electron-rich conjugated molecule can now undergo decarboxylation to generate product D, which is actually the plus 11 Dalton shift that we can observe here. Additionally, it can undergo oxidation and hydrolysis to now afford this amino substituted indole. And when we look at this particular peak under high-res mass spec, we observe that this is actually 1 Dalton different than this particular peak here. So we do believe that at this point, We have product C, the glycine conjugate, product D, the imine, and product E, the amino substituted indole. And we were able to confirm in this orthologous system that this reaction is indeed dependent on glycyl tRNA synthetase, glycyl tRNA, and glycine to produce this product. So at this point, we have one amino group on the tryptophan core. And so AMMB2 will add the tryptophan to the C-terminus. AMMC will trihydroxylate the modified tryptophan. And then we have oxidation to generate the quinone. AMMB3 can then generate this amino substituted indole. But now we can rationalize what the other pearls are doing in this system, as we have two left and we have two remaining amino groups on the molecule. So one of the two remaining pearls can then add glycine to the C-terminus of the peptide. AMM17, which was identified as a glycyl oxidase, can remove those two carbons to afford this product. Once we platomerize this product, it is now conjugated and has a vinylogous acid for which the remaining pearl can then also add a glycine, and it can undergo the same decarboxylation, oxidation, and hydrolysis to afford now the three groups on the product. AMM3 is a chlorinase that was identified by Brad Moore's group, so that can add the chlorine group that we observe here. Oxidation will generate a product for which now, once AMM12 or 19, the protease is encoded within the cluster, can cleave off the modified tryptophan, and the amino group here can now cyclase to generate the tricyclic structure that denotes the ammosamides. And now we're only a few steps away from the final product of the ammosamides. So in the broader context of the Pyrroloiminoquinones, we've discovered that these molecules, at least in the bacterial systems, are derived from post-translational modification of the precursor peptides associated within these clusters, and that the amino groups are installed via glycyl tRNA. And this is unique because most biosynthetic routes to aromatic amines are utilizing glutamine and glutamate. And the aromatic amines are often introduced prior to aromatization. So our system is unique in that some of the amines are installed before aromatization, and the nitrogen donor is different than the canonical groups that we see in most biosynthetic pathways. And so to summarize my work today, I've elucidated a new pathway to aromatic amines by the conversion of phenolic groups to aryl amines with glycyl tRNA as the nitrogen source. And this is a novel use for pearls than we have reported previously. We've also revealed a new paradigm in pyrroloiminoquinone biosynthesis in that these small molecules are made by post-translational modification. And so we've coined these natural products purlins, as they are the product of pearl biosynthesis. And finally, we've done enzymatic introduction of quinones to the C-terminus of peptides, and we could investigate this to provide a handle for bioconjugation. And I'm currently investigating the substrate tolerance for these enzymes. And so at this point, I would like to acknowledge my advisor, Wilfred Vanderdonk, for all of his guidance and support, all of the members of the Vanderdonk Lab, our funding sources, and thank you to you all for your patience and attention. And thank you to the organizers for giving me the opportunity to present in this seminar series.
Thanks, Paige. We're sending you a virtual round of applause for a wonderful talk and a really interesting biosynthetic pathway. Quite amazing. I think while people are putting questions in the Q&A, I'll go ahead and ask one myself. I was curious about the FMN-dependent enzyme that's doing a trihydroxylation, you know, and kind of like all over that tryptophan ring. So do you know anything about the sequence of the hydroxylations, or there's like a change in the binding event, or how you account for those 3 different hydroxylations at separate sites?
So I actually just came across a system in which I can trap it with one hydroxylation. So I'm currently investigating what the order of hydroxylations are. The enzyme was labeled as an unannotated protein in the sequence. And so when we subject this to all the bioinformatic analyses that we have in our pocket, we find that nothing really comes back, and there was no FMN-dependent binding site predicted for this protein. The only domain that has any prediction is a tetratricopeptide repeat, which is generally just for protein recognition. So a lot of investigation to do with the trihydroxylating enzyme.
Oh, very interesting. Thank you. All right, Anthony, do we have any questions in the chat?
There's none coming from them, so maybe oh, actually, we do have a question now, sorry. From Tristan Durand. So it says, great talk. Does it look like the free pearls in this pathway are derived from a duplication neo-functionalization event, or do they come from different ancestral pearls?
So we're also still investigating that. We're not sure whether they are derived from the LanB sequences. We're still investigating what came first. So that's another project that we're investigating in the lab. Another colleague of, sorry. I didn't realize the light would turn off if I did not move. And so that's a project that someone else is investigating in the lab to see whether these sequences are derived from ancestral pearls, or whether these pearls within the sequence of the ammosamide cluster are duplication events, but they all have wildly different sequences. The highest identity that any of the proteins within the ammosamide cluster share is less than 50% sequence identity. So these proteins are not very similar at all. So we're investigating a lot about the sequences and trying to derive what their amino acid specificity might be as well.
So maybe I can ask a question to follow up on your answer. So from you, your sequence alignment and a search for other pearls, how substrate specific do you think they are, and would you be able to? Do you think you would be able to identify other pearls with specificity for different canonical but also non-canonical amino acids?
Yeah, I think that eventually we will investigate the non-canonical amino acids. Thus far, we've identified cysteine appending pearls, tryptophan appending pearls, glycine appending pearls, and alanine appending pearls, and they all seem relatively specific for the amino acids that they add. When you co-incubate them with all of the amino acyl tRNA synthetases and their cognate amino acyl tRNAs, what we find is a very specific reaction in that it's only one product that we observe. So they do seem relatively specific for the amino acids that they add.
Thank you.
Okay, so we have another question coming through. So how widely distributed are pearls in terms of host phylogeny? How much diversity is there in the gene clusters where they're found?
Yeah, so there is quite the diversity. We've identified more than 600 sequences of pearls, and they range from actinobacteria to firmicute and several other organisms that we've identified. And there's a ton of diversity in the clusters. We observe 1, 2, 3, 4, 5, and 6, and 7 pearls in several different types of clusters. So we imagine that there are several different classes of natural products that are being biosynthesized by these systems.